Okay, hello everyone. Can you hear me? I think only. Okay. Okay, hello everyone. Good afternoon, morning, evening. I'm sure we're all coming from very different time zones at the moment. So thank you for joining us for this talk. This talk is part of the International Parkinson's Disease Genomics Consortium Africa webinar series. And so this is also in partnership with the Society of Neuroscientists of Africa, the African Academy of Neurology, and Trend in Africa. So I'm really happy to be here moderating this session. My name is Natasha Muanigwa. I'm a PhD researcher at the University of Luxembourg. There I study Parkinson's disease using human midbrain organoids. So without further ado, I'd like to introduce our speaker for today. I'm sure many of you know him or know of him. I'm just going to give a bit of a background about Dr. Andrew Singleton. First of all, thank you so much, Andrew, for being here with us. So Andrew received his bachelor's from the University of Sunderland and his PhD from the University of Newcastle upon Tyne. His research initially focused on the genetic determinants of dementia, in particular Alzheimer's, and in dementia with Lewy bodies. His postdoc was at Mayo Clinic in Jacksonville, Florida. He then moved on to the National Institute on Aging at the NIH and became a PI leading the molecular genetics unit in 2002. In 2007, he became a tenured senior investigator at the National Institute on Aging. In 2008, he became the chief of the Lab of Neurogenetics. And in 2016, he was named an NIH Distinguished Investigator. So we're very excited to have him today, and he's going to give us a talk on exploring the genetics of PD, the past, the present, and the future. And with that, I'm going to hand over to Andrew, and looking forward to hearing your talk.
Thanks, Natasha, for that really kind introduction. Although I have to say, it makes me feel kind of old. That list of stuff, really, yeah, wow. So thanks for inviting me to talk today. It's really nice to be able to share some of our work and what we've done over the years and where we're headed, both in terms of our lab and our consortium, and more broadly the community, the research community in this area. So I'll cover a few topics today. I'll talk a little bit about where we've been, where we are now, and also where we're headed to in the future. But if you think of topics, if you think of ideas, please write down your questions in the chat box, and I'll try and address them towards the end. And they can be on anything kind of related to this talk. If I don't touch on something, please just let me know. So I think this is a good way of thinking about why we do genetics, right? I'm a geneticist, so I think one of the key questions I have to ask myself every day is, why am I doing what I'm doing, and why do we work on genetics in general? And I think this is why most of us work on genetics. We're really trying to understand the beginnings of a disease. What are the underlying causes of a disease? With an eye initially, I think most of us initially did that to understand what the biology of that disease is. You know, if you can understand, if you can find a starting point or an early influencer in the disease process, you can then try to model those genetic changes and understand the underlying biology that really is the disease process, right? There's this series of biological changes that occur, and they're really what disease is, not the clinical presentation. If you can understand that, then it goes a long way towards finding a rational place for intervening in the disease process. Understand the biology, find places in that biology that you can change the process and either slow, halt, or maybe even reverse the disease process. In parallel with that, over the last few years, I think we've also started to think about using genetics, not just to understand biology, but to understand whether there are subtypes of disease. So we think of using Parkinson's disease as an example. I think most people in the general public, if you asked them what Parkinson's disease is, they'd say it's a disease. But perhaps it's several diseases, and there's a different biological basis for those several diseases that happens to end up in the same clinical and pathological entity. So not only can genetics tell you about the biology of disease, it can also tell you about whether there are subtypes, and on top of that, start to tell you about, or start to provide information about who's going to get disease and what type of disease they get. And I think those three things together are particularly important, because whatever treatment we come up with, almost whatever treatment we come up with, it's going to be really important to be able to apply that treatment extremely early in the disease process. The earlier we can apply the treatment, the less effect it's going to have on the patient's life. And actually, I think the more chance it has of being successful. And the other part is, of course, we want to match the patient to the treatment. So we need to understand what the biological basis of an individual's disease is, so that we can then attempt to address that biological basis. So with that in mind, with this structure in mind, I thought I would tackle a few different things. I'll talk about the genetic architecture of risk. I'll talk about modifiers of not just disease, but things like age of onset and of different genetic forms of disease, right? So we have some diseases that have a single large genetic risk factor, but there are influences of those types of disease. And I'll talk about some efforts to understand those. I'll talk about something that I think is really is important in the context of how we're changing how science is done. And that's around generating large resources and large tools. So I think that one of the things that we've seen over the years is that science has progressed a little from small individual labs working on discrete problems to large consortia, large groups of individuals working to address a very large problem. And part of that is in generating resources and generating tools. I'll then talk a little bit about the future and where we're headed in the future. So genetic architecture of risk. So before the 1990s, I think we thought Parkinson's wasn't a genetic disease. And I think if you went to med school before then, you would probably be told that Parkinson's was the typical non-genetic disease. That changed in the mid-90s to a certain degree with the discovery of mutations that cause really rare forms of disease. And in Parkinson's disease, as in many other diseases, the mainstay of genetic research was around finding extremely rare families where disease occurs more often than you would expect by chance, and looking for a causal mutation, something that if you carried it, you had a very, very large chance of getting disease. So 1997, the discovery of synuclein mutations was really the first stepping stone in PD genetics research, followed a year later by the discovery of Parkin mutations, and then a whole series of discoveries, DJ1 mutations, PINK1 mutations, LRRK2 mutations, et cetera, et cetera, et cetera. Now there are somewhere in the region of a dozen or so genes that contain mutations that confer a very high risk for disease. I'm going to talk about something that's slightly different. For the most part, most of those genes are fairly rare causes of disease. There's one exception, which is LRRK2, which contains common-ish mutations in the general population, but generally they're quite rare. So I'm going to talk about genetic investigation of the rest of Parkinson's disease, the other 95–96% of disease, which is that disease where there isn't a strong family history. And I think that for a long time we mistakenly thought that no strong family history meant no genetics, and they're different things. What no strong family history means is that there's no simple genetics. It doesn't mean there's no complex genetics, and that's what we're going to talk about today. Complex genetics, where an accumulation of genetic risk factors imparts risk for disease. So the first thing that you really want to know, if you're starting to try to understand the basis of a typical apparently sporadic disease is how much genetic influence is there? Is there any genetic influence? And how much should I be looking to find? Basically, when can I stop? When can I stop looking? So this was typically done with twin studies, or where you would look for at monozygotic and dizygotic twins, and you would look at the ratio of sharing of a trait, in this instance, Parkinson's disease. So if the disease was purely genetic, you would expect half of dizygotic twins to share a trait like Parkinson's disease. So if one had it, then 50% of the time, the other twin would have it. Whereas monozygotic twins, you would expect sharing to be complete. If one twin had disease, the other was sure to get it. So some early work was done in this area. In Parkinson's disease, probably 20, 25 years or so ago. And it showed there was a genetic component, but a fairly minor genetic component. There's lots of kind of issues with those studies. They tend to be very small. Of course, you have to find twins. You have to track them over a long period of time, so they're not always that accurate. So one thing that's come along to address this challenge to understanding whether there's a genetic basis is this technique called complex trait analysis. And this is a way of looking of sharing of genetic information. So you can take a very large population of people with disease, a very large population of people without disease, and you can look at how related each individual is in those populations. And this is related at a very, very distant level, generations and generations and generations before. So even in an outbred population, you can look at this type of sharing. And if you look at the sharing, the genetic sharing in the cases, and you compare it to the controls, that can give you a proxy for what genetic influence there is in disease. So if we do this in Parkinson's disease, what we see is that Parkinson's disease has a heritable component of about 30%. So another way of thinking about this is that the average Parkinson's disease case is 30% genetic. There's some other stuff going on, or 30% heritable, I should say. There's some other stuff going on, whether that's environment or genetic changes that are not necessarily heritable or difficult to track. When we started to try and identify what the basis of these genetic changes was, 15 years ago, 2005 was the first study, we had limited success, and that got better and better as time went on with increasing sample sizes. So we're at the stage, about 2 years ago, we're at the stage where we can say we know about 30% of that 30% of heritable component of disease. So about 10% of the total risk for disease, and only about a third of the heritable component of disease. So this tells us that there is a substantial amount of heritable risk for disease, and that we've still got quite a lot left to find. So how do we find it? There's lots of different ways. The most prominent and the easiest, I guess, in the first instance, is to increase sample sizes. So to do genome-wide association studies, where you're taking very large populations of individuals and doing a survey of genetic changes across their genome, and looking at the differences in the proportion of those changes in cases versus controls. So this study was published last year by our group, and to give you an idea of scale, this included somewhere in the region of 50,000 cases, something like that, and about 1.4 million controls. So we got 50,000 people with PD, or a first-degree relative with PD, and then 1.5 million people without disease. In each of those individuals, we're then looking at about 11 million genetic changes in their genome. So we're typing them for 11 million genetic changes in their genome. And basically, although the data is large, doing something very simple. At each of those 11 million sites, we look to see the frequency of that change in cases versus the frequency of that change in controls. So what this gives you is a difference in allele risks, and and an ability to look at p-values between at each of these sites. So it gives you significant regions of the genome, areas in the genome that contain risk for disease. Okay, so sorry, I'm just getting a message about muting. So what this gives you is a plot like this. This is called a Manhattan plot. It's supposed to look like the skyline of Manhattan, where each individual dot is a variant, and they give us a p-value for whether that variant is associated with disease or not. And you can see these peaks all the way throughout the genome. This is 11 million SNPs throughout the genome, and those that are significant with a p-value above 10 to the -8, so a very, very low p-value, are labeled in red. So we have these 90 or so regions that contain risk variants for Parkinson's disease. And this really is a tremendous advance. We've gone from a space where we had absolutely no genetic risk factors for disease 15 years ago, for Parkinson's disease 15 years ago, to one where we have 90 and we have a path forward to finding more. So you can do lots of things with this data. You can do things like project risk. So you can accumulate all of these data together and look at genetic state in an individual and predict what their risk for disease is. You can also now start to look at you can make functional inferences from this genetic information. So the very kind of first wave of doing that was to look at these genetic changes that you identify as risk factors for disease and see whether they have any immediate biological effect. And the most common biological effect we would look at was gene expression. Does this variant that imparts risk for disease also change expression of a nearby gene? With the idea then that you can say, well, what we think the biological mechanism is expression of this gene. This gene is overexpressed or underexpressed, and that is the influence. That's the thing that's affecting risk for disease. And we see that in about a third or so of the risk loci that we've identified so far. So about a third of these risk loci also affect gene expression in the human brain. But this is pretty kind of gross look at expression. It's taking a chunk of brain from 100 different or so brains and looking at expression in the total mass of cells that you've got there. You can also start to do things like infer what tissues are important in a particular disease. So if you have your risk loci, you know all of the genes that are under those risk loci, and you can look at where those genes are most expressed and most regulated. And then if you have an atlas of gene expression across tissues throughout the body, you can rank those tissues to say which are the tissues where the genes of interest are most regulated and most expressed. And you get a plot kind of like this, which makes a huge amount of sense. What it's telling us is everything in red here is brain, everything else to the right are other tissues. So this tells us, as you would expect, that Parkinson's disease genetic risk is mediated through brain tissue, not through peripheral tissue. A fairly newish technology has allowed us to take one step further, which is single cell sequencing. So before, as I mentioned, all of the expression work was done in chunks of tissue, and we were looking at a gross signal, a signal of everything in that tissue block together. Now we're able to do single cell sequencing and look at individual cellular populations. So when we do exactly the same experiment, look and see at where our genetic risk genes are expressed in a cellular context, what we see is something really interesting. Again, perhaps not surprising, but pretty interesting. And that is that the space in which our risk genes are most exquisitely expressed is in neurons, and of those, substantia nigra and globus pallidus neurons and dopaminergic neurons are the ones that shine up the brightest. So this is really important. I mean, I think there's been a lot of discussion over the years about what's the important cell. I mean, I think that, of course, we all thought it was dopaminergic neurons in the nigra because those are the neurons that are lost. They're the cardinal hallmark of disease. But this is really unbiased proof that that's the case. So this tells you when you're modeling disease from a genetic perspective, the cells you should be looking in, or at least one set of cells that you should be looking in, is dopaminergic neurons. They're the cellular context for this genetic risk. So I've given kind of a short overview of genetic risk for disease. What about genetic risk for other things? So other facets of disease, not just whether you get it, but when you get it. So of course, we can look at genetic risk for age of onset. And this is a study, again, done by our lab and the IPDGC, led by a guy called Cornelis Blauendraut, who I've worked with for a few years. And essentially what you do here is, rather than look at the genetics of cases versus controls, you look at the genetics just in cases, but the outcome that you use, the thing that you're trying to associate genetics with, is their age of onset. So here we have 28,000 cases split across a discovery and replication cohort. Same kind of genetic work. You look at 11 million or so variants and look for their association with disease. And what you see is something a bit flatter than the last plot, in part because it's much less, the sample size is much less. The other part is that the phenotype is kind of fuzzy. For PD, you can be pretty sure someone's got PD. Age of onset, it's never really clear what the age of onset is for an individual. So we don't see as many hits here, but I think this will improve over time. We see 3 genome-wide significant things associated with age of onset. SNPs at the synuclein locus. Basically, if you do any genetic work in Parkinson's disease and synuclein doesn't show up, then you've probably done something wrong. It shows up in everything. A hit at a site called TMEM175 GAC, which is also a risk factor for typical PD. And this site at ApoE. I'll just say that ApoE is a red herring. It's associated with just age. It's not age of disease, it's just age. And that's, of course, because it's a big risk factor for Alzheimer's disease. So the number of people that carry it declines over time as they succumb to the disease. So what's interesting about age of onset, I kind of expected all of the hits that we see for risk of disease to show up in age of onset for disease. And by and large, they do, but there are some exceptions, and I think those exceptions are fascinating. So if we look at the effect size, if we look at the association effect between age of onset on this axis, on the vertical axis, and then just risk on this horizontal axis, you should expect a straight line. If the association for age of onset is exactly the same as the association for risk, you just expect a straight line. And what we see are some outliers. So tau, the tau region on chromosome 17, is a major hit for risk for disease, for Parkinson's disease, and it's completely flat for age of onset. I think this is fascinating from a biological perspective, because it tells us that whatever is going on at that locus, and we don't know what is going on at that locus, but whatever is going on there is affecting whether you get disease, but not having any influence on when you get disease. And that's a really interesting biological phenomenon when you think about it. So far, all of the other risk loci that we've looked at, the more of them that you carry, the younger you get disease. And this just isn't the case with tau. So I think there's some really interesting biology going on there. What about other genetic forms of disease? So I mentioned there are a bunch of what we sometimes think of as monogenic forms of disease, where if you carry a mutation, you have a high likelihood of getting it. There is also a gray zone. There's this area of mutations where if you carry them, they have very high risk they're rare, but they have very high risk, and you almost certainly get disease. Then there's the things that we've talked about so far, which are down here. These are variants that are common in the general population, but they only increase your risk a small amount. There are things in between, one of which we're going to talk about next, which is GBA, which is relatively common. So variants in GBA are fairly common, and they impart moderate risk. So they increase your risk 2 or 3-fold. So a way of thinking about that is if in the general population, 1 in 100 people get PD, about 2 in 100 or 3 in 100 GBA carriers get PD. As you know, carrying 2 mutations in GBA causes Gaucher's disease. Carrying a single mutation is a risk factor for Parkinson's disease and dementia with Lewy bodies. So the question is, a very large proportion of people who carry GBA never get disease. It certainly increases their risk, but they never get disease. And we wanted to know, is there a genetic component to that risk? Is there a genetic effect on that risk? So essentially you can do something along the lines of what I've presented already so far, which is you can look for, you can use genome-wide association to look for risk factors that alter whether you get disease when you're a GBA carrier. So in this instance, we looked at around 2,000 cases who have a GBA mutation and a few thousand controls who have a GBA mutation but don't have disease, and look for other genetic differences between those individuals. And again, you see synuclein every time, but you see a couple of other hits, this hit here, CPT2 and cathepsin B here. What's particularly interesting about cathepsin B is cathepsin B showed up as a general risk factor in PD. It's a lysosomal protease. So it's involved in proteostasis in the lysosome. And of course, GBA is a lysosomal storage disorder. So there's a nice clean biological link there in that CTSB increases risk of disease in GBA carriers. Now, I'd mentioned earlier that CTSB is just a general risk factor in PD as well. So we're doing the same thing here we did a few slides ago. We're looking at the effect size of risk SNPs for PD versus the effect size of risk SNPs for PD in people who carry a GBA mutation. And again, we see some outliers. We see generally a straight line, but there are some things that have a much greater effect, and CTSB is one of them. So the CTSB risk allele has a much stronger effect in GBA carriers than it does in the general population. So this is really it looks like a true gene-gene interaction, and this is the only one that I've ever seen. But again, it tells you something about the disease that, that is pretty interesting. And that, I think, is that there is potentially a mechanistic subtype, or not necessarily a subtype, but a subcategory here, something that overlaps with the rest of the disease, but is also subtly different mechanistically. And that difference is driven by CTSB and GBA interacting in some way, shape, or form. So there's a fair amount of work left to be done in this, including some additional replication. But again, I think this is starting to give us the first hints about subclassification of disease. And going back to that first chart that I showed, where we're attempting not only to understand the biology of disease, but also to subclassify disease and match therapies to individuals. So what about tool generation? Again, I mentioned that a lot of the stuff that we do tends to be very large, very collaborative. If you look at the GWAS work that I talked about, there are 200, 300 authors. So we're used to doing things in large collaborative groups. And I think we need to continue to do that. And this is one of those groups, although a little smaller than most genome-wide association studies. So this is a collaboration that we formed called Found in PD. And the idea here is to generate foundational data in Parkinson's disease. And the reason we need to do that is because we've become very good at finding genetic risk factors for disease. And although we still have quite a few left to find, the path forward is pretty straightforward. We kind of know what we need to do genetically to find them. What is less straightforward is taking those genetic risk factors and understanding biology. So this is a study that aims to produce really pilot data to try and inform biology from a genetic perspective. So this is the loose methodological design. We've taken 150 or so IPS lines, induced pluripotent stem cell lines, from well-characterized Parkinson's and control patients. So these are patients that we have complete genetic knowledge about. They've had their genomes sequenced, and we have a ton of clinical information about. We then take those lines, we push them towards a dopaminergic neuron lineage, because we know that dopaminergic neurons are at least one of the cell lines that we should be interested in. And then we measure a whole bunch of different things. We look at epigenetics, so we look at DNA methylation, how open or closed or how accessible the DNA is, which is a proxy for transcriptional potential. We look at things like high C, so this tells us which parts of the genome associate with each other. So it's incredibly important to understand how the DNA loops together and which parts interact with each other, because that tells you about how regulation occurs and how a piece of the genome that's 200,000 base pairs away from a gene affects that gene. We've looked at proteomics, we've looked at expression using RNA sequencing, and we've also done the same using single cell techniques. So when you do an IPS study like this, you're trying to produce dopaminergic neurons, but really only about 20% of your cells end up being dopaminergic neurons. So rather than just take all of the cells and hope that your signal from this minority shows through, what you can do is do single cell sequencing, and you're able to define what the cell types are, but also get biological information about those cell types. So this data is complete now and will be publicly available in the next 2 or 3 months. So I'm hopeful that this kind of resource project will be useful for the general population. You'll be able to go to a website. This will be a website available through the Michael J. Fox Foundation and the PPMI study. And you can get any information you want from these 100 cell lines in terms of gene expression or genetic contribution to disease, or what their DNA methylation looks like. So it really gives a nice starting point for understanding the basis of disease. And here I'm just showing some of the browsers that we've got here. So for example, you have a browser that shows all of the genome-wide association peaks, and you can zoom in on this area and it will tell you which parts of the rest of the genome associate with this area, what expression looks like, what expression of the genes in that area looks like in dopaminergic neurons or other neurons or astrocytes or glia. So really, again, giving us insights into biology. And here's just some single cell data, which I won't go into. This is a large amount of data and a lot of collaborative work. One of the things that we are trying to move forward with, but it won't be in the first iteration, is to enable anyone in the world to be able to analyze this data in depth. So at the moment, or rather I should say in about 2 or 3 months, a browser will be available. So if you're a researcher interested in from anywhere in the world, you should be able to log into the browser and really just look at the data. You can manipulate it a little bit and put in small parameters to investigate the data, but that's about as far as you'll be able to go. The next stage that we would like to really develop is to enable in-depth analysis from anywhere else in the world. And of course, the cloud is great for this. We can have a whole series of machines that store the data and analyze the data in its place, and you can access those machines from anywhere in the world. So more to come in that area. So what about the future? I'm going to talk just for another 5 minutes or so. Let me see, what time is it for? Yeah, about 5 minutes or so on this initiative, which is the Global Parkinson's Genetics Program, because I think this is a really exciting initiative. This is something we're leading here, but that is, I'd say the leadership is really much more broad. It's an initiative that aims to bring together the Parkinson's genetics community into one space. So although I think the genetic community has been generally quite collaborative, there are still some silos and some places where people haven't been able to interact too easily together. And the idea of GP2 is really to bring everybody together into one space and to work together globally. The other part, which I'll touch on a little bit later, is that in Parkinson's, like in most disorders, we've had really good success in looking at risk for disease and characterizing the genetics of disease in Northern European ancestry individuals. And that's because the samples are available and easy to get to. What we've been really terrible at is looking at the genetic basis of disease in populations outside of Northern European ancestry. There's been a little bit of work in some populations from Asia, particularly from Japan and China, but really no, it's been limited for everywhere else, and that's something that we're aiming to change here. So this is a study that is sponsored by the Aligning Science Across Parkinson's Initiative and the Michael J. Fox Foundation. And they've come up with these themes that they want to support around understanding the biology of PD genetics, looking at immunity, understanding circuitry. But underlying this is a need to create foundational data. And the GP2 program, the Global Parkinson's Genetics Program, is part of that foundational data. We need to produce foundational genetic understanding of disease in diverse populations in order to be able to understand biology, in order to understand progression, in order to understand lots of different things. So the overall goals of GP2 are to have a really dramatic expansion of our understanding of the basis of PD. And to do that, we have to create a truly global network of functioning investigators, scientists, clinicians, all working together towards that end. I think a critical part of that, and one which we can talk about, is providing training and opportunities for folks around the world. So the deployment and the availability of resources is different in different places, and I think that leads to a disparity in opportunities for folks around the world. And part of GP2 is to try and even out that playing field a little bit, to not only make data available, but to help build the expertise and provide the resources in order to be able to work on that data. So we're really very focused on democratizing data. So not just making it available, but actually giving people the tools and the skills to work with it. So in a bit more detail, mapping the genetics of individuals with Parkinson's disease. So this is both typical Parkinson's disease, but also rare familial forms of Parkinson's disease in Northern European ancestry individuals, in African Americans, in folks from Africa, from sub-Saharan Africa, from Southeast Asia and China, India, and South and Central America. So really a global initiative. We'll work with all of those groups to produce really rigorous genetic data on each sample that's provided to us, and to push that data back out to the research community through this data ecosystem that we've talked about, all with the idea of creating a foundational resource that can be used by the global research community, and being open, getting feedback from investigators, understanding what investigators' needs and desires are, both in terms of how they interact with the study and what they'd like to do with the data. This is just the organizational structure. So you've got the kind of things that you would expect around understanding operations and compliance, because compliance is so different around the world. You know, legal requirements for sharing genetic data and sharing genetic material are very different in one country to the other, and we're trying to navigate how to do that. We also have groups working on complex disease, groups working on monogenic disease, and groups working on trying to help us understand what the needs and opportunities are in underrepresented populations, and how to support those needs and populations in a thoughtful way. So we'll be genotyping 150,000 individuals, cases. We'll be generating whole genome sequence, so full sequence on around 10,000 individuals, and that will include unsolved monogenic cases, you know, cases where there's a strong family history, but we just can't figure out what the cause is. But also populations where there isn't good reference data, where there's not good existing genetic data. We'll also be producing some long read sequence data. This is quite a new technique that allows us to look at some of the hidden parts of the genome that we weren't able to look at before, or able to evaluate before. So things like large structural variation and repeat expansions, parts of the DNA that expand and contract. So that was the talk, but I'd been asked to talk just very, very briefly about my journey to the kinds of science I'm doing now and how I got there and all those kinds of things. So I thought I would start by telling you, and I'll keep it short because I know this isn't that interesting, but where I grew up. So I grew up right here on a place called Guernsey, which is, if we zoom out, a tiny island in the English Channel. So in between England and France, much closer to France than it is to England. There's about 50,000 people there, very small society. You kind of know everyone on the island. You know, it's very isolated upbringing, I would say. I moved, as was mentioned earlier on, to a place called Sunderland in the UK to do my first degree. So this is in the northeast of England, right here. And then I did my PhD somewhere about 30 miles north of there in Newcastle. So I went from this tiny island to a smallish city. I got to experience a bit more, to a larger city in Newcastle. And each of those things were an incredible growth. You know, I got to learn new things about myself, new things about science. My scientific horizons really changed in a big way, I would say, when I moved to a place called Jacksonville in Florida. So here, across the channel, from all the way up over here down to here to sunny Jacksonville, Florida. I worked at a place called the Mayo Clinic for a couple of years, trying to understand Parkinson's disease. And really the change there was a change in resources. Where I worked in the UK, we had kind of limited resources, limited capacity to do stuff. And in America, there were a lot of resources, and you know, you could really ask really cool research questions. After working there for a couple of years, I then moved up to here, which is Maryland, which is where I am now. I've been here for 20 years or so. I'm working at the National Institutes of Health. So this is the government-sponsored research center, has about 20,000 employees, all working on various aspects of medical research. Really an incredible place. If you don't know how to do something in terms of science, someone there does. It's such a big place. One of the things that I think that's been really great and enjoyable over the last 20 years in my research career has been people. So I'm an introvert, right? Generally, I don't like people. I'm shy. I like to keep to myself. But one of the things I've really been able to do and enjoy to a certain extent is to travel all over the world and meet with clinicians and scientists all around the world who are working on various problems, and to understand that actually we're all working in the same way on the same types of things, and our motivations are all the same. We're all trying to solve these really compelling problems and to try and give back and give something to our general communities. I think that in a way has shaped the type of science that we do now. So again, 20 years or so ago, a geneticist would be in a small group of 2 or 3 people working in a lab by themselves on a problem, competing with everybody around the world, and getting an appreciation that we're all kind of trying to do the same thing. We're all trying to get towards the same end goal has really helped me understand that the path forward, the route forward that we have to have is to work together and to work collaboratively. And that's why we're in a space now that we're in, which is working on these very, very large projects where you have to have large groups of people and creating community resources and working together as a large group. And that's been a really fun journey for me. I've gone from a small place physically to one where I've gotten to learn more and more things as time has gone on about people, about science. And I think that's mirrored the type of science that we do now, from small science to large science. So it's been a really fun journey. I would say the other really cool part about this large science is that it creates lots of opportunities for lots of different people. And particularly now in the context of being able to access remote data and do things remotely, those opportunities have never been as great. There really are a tremendous amount of opportunities to work on scientific questions, no matter your background or location. So with that, I'll stop talking and we can take questions and have a chat if that would be helpful.
Yes, thank you so much, Andrew, for that talk, first of all, about the science, but also going into telling us the GP2 consortia. I think for many people who are probably joining on this, for this talk today, a lot of them are from Africa, actually, and I think the fact that we now have this consortium that will also be doing work to study the genetics in other populations, I think that's really, really cool. I think it's super important. And I think a lot of us know that a lot of populations haven't really been well represented historically. So I think this is such a great initiative. And I really liked hearing about your journey as well, particularly what you said about the change of resources that you experienced once you moved over to the USA, because I think that also goes to highlight the importance of, making sure resources are available everywhere so that we can have quality research happening and we can increase the amount of knowledge that we can gain across the world. So thank you for that. That was really a great talk.
Yeah, sorry, just to touch on that point, it's interesting actually, because I think that there were limited resources where I started and where I did my PhD, but there's a real advantage to that in some ways. And that advantage is it makes you think very, very carefully about experimental design and about whether the question you're asking is the right question to ask, so that when you do come across resources, you can really take advantage of them, right? Because you're thinking carefully about science all the time. So yeah, I think that training in that space is really useful.
Absolutely. So I just want to remind the participants, if you have a question, please type it into the Q&A. I think a couple of people raised their hands, but just for the sake of it not being confusing, just type in your question in the Q&A and we'll get to it. There are a couple of questions that were already posed. I'm just going to try and go back on some of them. So there was one question from Jonathan, and he said that you referred to subclassification, I guess subclassification for PD. But what do you do about the inaccuracy of the PD diagnosis? Isn't the accuracy of the phenotype critical to interpreting genetic data?
Yeah, that's a very good question. So I'll answer that in a couple of ways. So yeah, I think I'll answer that in a couple of ways. So in terms of diagnosis or classification during diagnosis, it is true for sure that early on in the diagnosis, there's a fair amount of inaccuracy. And we've been working on some models to help try and improve that accuracy. And it turns out you can do it with fairly simple data. So a combination of genetics, so you take all of the genetic risk factors and you create a genetic risk score. When combined with smell deficits, anosmia, age of onset, sex, and family history, you can classify someone as a case or a control with about 98% accuracy. So that works. That worked pretty well. I suspect, though, that the question was a bit more about clinical subclassification, and how, so when I was talking about classification, I'm really talking about biological classification. So not whether the disease looks any different, but whether the biological basis for the disease could be different. And I don't think necessarily that a biological subclass is going to relate in a different clinical subclass. They might do, but those are kind of separate questions. I will say, however, that one of the things we've been trying to work on, and this is outside of genetics actually, this doesn't include any genetic information, is whether you can predict clinical course based on presentation at baseline. And this, there's a kind of a cool paper by a guy called Faghri, Faghri is his second name, that uses machine learning and artificial intelligence to try and create subclassifications of disease in people for whom we have longitudinal data, clinical data, and then go back to baseline and say, could we predict those subclassifications? And it seems to work quite well. So I think whilst it's early days, I think in answer to Jonathan's question, I think it's going to be a combination of clinical acumen and collection of standardized data that we can use advanced computational methods on. Sorry, that was a lengthy answer.
No worries, we like thorough answers. So another question coming from Sophie. She said, great talk, thank you. And she's really excited for what is to come, especially with found in PD. So you mentioned that in the analysis of the tissue expression, the most significantly enriched tissues were all brain derived. Do you think the peripheral tissues could be contributing to disease risk, given many studies have shown alpha-synuclein pathology in multiple peripheral tissues?
Yeah, I think so. Yes, I think so. Whether it's going to be peripheral nervous system or cells associated with the nervous system, I mean, one area that's been under increasing investigation is the role of inflammation and immunity, right? So there are some genetic hits that don't clearly relate to dopaminergic neurons, like the HLA locus is a great example, right? So I think that there is a role for immunity in there somewhere. I don't know how just yet. I think it also makes us start to ask a question about the types of things that are being found in genome-wide association studies. Are they things that initiate the disease, or are they things that are affecting response to some kind of pathogenic stimulus? And that I think figuring that out will tell us something about the biology of disease and the role of peripheral tissues.
Yeah. Another question coming from Yared. So with the current understanding that PD is a genetic disease, can we still say PD is idiopathic? How does that affect our clinical practice in the future? Do you think we will run genetic tests as initial workup of PD cases anytime soon?
Yeah, that's a great but tough question. So PD is absolutely a genetic disease, but it's also something else too, right? Genetics doesn't explain all of the disease. And I think that we got stuck for a long time in thinking about genetics versus environment, when the answer is genetics and environment, right? They have to interact in some way. And I think this is probably true for all late onset diseases. You have genetics, which kind of gives you this foundation for disease processes, but other stuff during life has to affect that. whether that's environment or actually even just chance. You know, and here I'm thinking about molecular chance, collisions of molecules leading to formation of a dangerous species. I think those things have to interact. Do I think there's a role for genetics now in working up patients? Yes and no. I think it depends on the patient and depends on the type of genetics. I think an answer 5 or 6 years ago would have been it's only useful for things like family planning, for instance. Let's say you wanted to know your genetic status before you just started to have a family. For most people, that's not an issue, but some people might be interested in that. It doesn't really change your treatment yet. Genetics, your genetic origin doesn't really change the origin of your disease doesn't really change your treatment yet. I would say that I think that perhaps not clinically, but societally, that is changing a little bit in that people now just want to know whether there's anything they can do about it. Services like 23andMe have made people curious about their genomes and their genetic risk and yeah, and whether it can impact on their lifestyle or not. So, you know, if I have PD and if I have a high number of risk factors for PD, should I start drinking coffee? Because coffee is perhaps protective against PD. There's some suggestion of that. So I think the answer is it depends on the individual. But in terms of clinical practice, it doesn't affect things too much right now.
So I actually have a question myself. I was wondering what you think. So there's been talk for some time now about this neurodevelopmental hypothesis that Parkinson's disease might not just be neurodegenerative, but might also be neurodevelopmental, because if, let's say, you're born with these genetic risk variants, or you're born with one of the like LOCK2 mutation, synuclein, etc., that you're already born with the mutation. Do you think that being born with these mutations already affects the way that the brain or the substantia nigra is developing, like all throughout life, or you need that context of the aged brain to actually start having the PD associated changes?
Yeah, that's a great question. I don't know the answer to it, to be honest. I mean, I think that you have all of these functional alleles. If you carry a lot of genetic risk for PD, you have functional alleles that are affecting things, and they're affecting things throughout your life. So one would assume that they're having an effect on development. Whether it's a large effect, whether it's a disease-associated effect, I don't know. Yeah, that's a tough question, and I think it actually relates to the type of work that you're doing, Natasha, which you know is around, should we be looking at very mature neurons or very mature organoids, or is it okay to look at things in developing state? Tough question.
Yeah, absolutely. I asked because it's something we discuss a lot, because in organoids, even in very early organoids, you sometimes see things. So you wonder, is it of what you see in the brain, or is this just also because it's an artificial system? So I think there's a lot to still be understood there. Yeah. So there's a couple more questions. I think this is quite an interesting one, and given the current what's going on in the world. But someone asked, as we all know, that COVID-19 is associated with anosmia and dysphagia. I think that's how you say it. Does this give us any clue about PD pathogenesis? Would we say they might have an increased risk of PD in the future? I think it is interesting because there have been some reports about the neurological impact. Maybe you can comment on this.
Yeah, I mean, my answer is going to be a complete guess, right? As everyone's is. I don't think the anosmia is going to tell us too much about biology, because the anosmia is reversible, right? And that's not the case in PD. Whether it's going to have an effect on disease long term, I just don't think any of us knows, to be honest, right? I mean, we're all just kind of guessing. Yeah, I don't know. I hope not, but I simply don't know.
yeah, you're right. I don't think anyone at this point can
Yeah.
think of an answer to that. So maybe we'll take just a couple more questions. So there was one, again from Jonathan, where he asked that, do you think it's likely that 10 or 15 or 20 variants contribute substantially individually by acting on one another to make a critical difference to crossing a threshold to develop PD over a lifetime? If so, that is unfortunately likely to be very tricky from a therapeutic perspective. Does this effectively amount to a bad set of cards that one is dealt with at the time of conception?
Yeah, yes, I think. So we know that cumulatively, the cumulative genetic risk affects disease. So they're kind of mostly additive in a way. If you take these 90 risk loci, they're mostly additive. Each one that you carry adds a little bit more of your risk for disease, which takes you towards this threshold for disease, or primes you for disease in a way. There are some potential exceptions, one of which is the GBA TMEM exception, where they don't kind of add on top of each other, but they synergize, and the whole is greater than the sum of its parts. I actually am not too worried about what this means from a therapeutic perspective, because actually I don't think necessarily the genes we identify are the therapeutic targets. I think the networks that they affect are the therapeutic targets. So it actually makes me feel quite optimistic about therapeutics, because the more genetic influence we understand, the better we can track down those networks that are the disease, and find nodes in the network that are worth intervening with therapeutically. So I actually think the future is quite exciting in that area.
Yeah, that's very encouraging, I think, for us who are working in that space, because indeed, I think not necessarily targeting the gene, but the pathways or as a whole that are affected may be the way to go. So I think for the final question, I will take it on a different note. So there was a nice question from Sarah, which she asked, what's your advice to young graduates who have admired your journey in research? I think just generally your advice in navigating the space of research, and maybe some things that you can just share with us that maybe you wish you would have known when you were at our stage earlier on in your career.
Yeah, I think don't be shy. That's the first thing, right? Science is, we were talking about this before the seminar started. We were talking about COVID, and how one of the challenges is that it's restricting interaction, that casual interaction. And that is so incredibly important in science. You have to make connections with people. And I know that early on in my career, and even today, I am a little bit intimidated when I meet people, and shy to talk to them about science. And you have to get over that, even if it's uncomfortable. If you're at a meeting and there's a big shot there, and you just have to summon up the courage and go talk to them, and talk to them about your research, talk to them about your ideas, because generally people are nice and they're interested in what you have to say, and they're interested in hearing your story. And science, like so many other things, is about connections. You have to make connections in order to progress. You have to meet people. You have to interface with people. I think the other thing I would say, what's worked for me is to work with good people. So when you meet someone, you can tell whether they're a good person or not pretty quickly. As soon as you establish that, make them someone you're going to work with, because those are the people that are going to look out for you, make sure that you're able to progress, and actually make it fun. No matter how driven you are, if you come to a job every day and it's not fun, it will kill you in the end. So, work with people that you enjoy working with, and that will help you in your enthusiasm and your path forward with science.
Thank you so much for for that. Thank you for the talk. I'd like to thank everyone who participated, who joined us today for the seminar. There are many more of these coming up in the coming weeks, so please keep up to date. I think there's a link that was just put in the chat, so you should copy paste and save that so you can keep in touch. But yeah, so I just want to wish everyone a great day, evening, morning. I'm not sure what time zones you are in, but enjoy the rest of your day, and we will hopefully see you again next time.
Have a great day, everyone. It was lovely to talk to you.